Hello everyone, my name is Mariam Nasir and I am from Department of Molecular Biology and Human Genetics. So my advisor is Professor Ming Derlin and my co-advisor is Professor Izulin. So I rated myself 80% as I have completed the preparatory phase and the experimental workflow is established. The remaining work involves advanced experiment schedules for the next stage. This is my progress report checklist which my professor signed and also checked my progress report. These are my list of the courses that I took in the first semester. So the total credit hours I take in this semester are 7 credit hours. And this is my safety, health and laboratory training certificate. So these are the course details. I successfully made up the classes due to 2 weeks late arrival, completed all required presentations and course work and submitted final presentation and assignments in accordance with the course requirements. Throughout this semester, I have the opportunity to learn and explore various subjects and topics, and I also gained valuable communication skills through presentation, discussion, and written assignments. I also performed practical lab work on small scale partition cell culture, which include thawing cells, cell counting, cell seeding, and thin layer chromatography. So I arrived at Suchi University on 20 September 2025 and then joined my co-advisor Professor Izurin lab on 22 September and 23 September I started to work with my co-advisor to learn the experimental techniques used in the lab and presentation preparation for the seminar. This is my research project titled Chemical Analysis of Bioactive Natural Products with Anti-Osteoporotic Potential. Basically, we want to check the effect of compounds from the natural plants that are used against the osteoporosis. This is the overall experimental design of my project, which starts from the large scale extraction of the EIG, followed by the different solvent partitions into hexane, ethyl acetate, and butanol and water fractions. These fractions are first evaluated in vitro using the bone-related cell models, a Rankel-induced osteoclast cell model, and primary human osteoblast cell model to assess different type of assays. These fractions which shows positive effect will then be further tested in vivo using an oviprimine induced bone loss mouse model which shows postmenopausal osteoporosis, and bone changes will be analyzed using the micro CT biomarkers and histological staining. Overall, this design allow comprehensive evaluation of both bone formation and bone resorption, helps to determine the anti-osteoporotic potential of plant extract. At the bench work that I learned during this semester that includes the thaw cell culture, different cell culture techniques, and the small scale partition of the plant samples and their thin layer chromatography. This slide shows my cell culture practice and experimental preparation. I repeatedly cultured and subcultured raw cells, monitored their growth under the microscope, and performed cell counting to ensure the healthy and consistent cell density. This practice helped me improve my technical skills in cell handling and prepared me for upcoming cell differentiation and functional experiments. This shows the small-scale solvent partitioning of the plant extract. First, 1 gram of crude extract was dissolved in 50 ml of water. The aqueous extract was then sequentially extracted with different solvents of increasing polarity. Hexane was added 3 times and the hexane layer was separated and dried using the rotary evaporator yielding 206 mg. Next, ethyl acetate was used in the same way, producing 110 mg yield. This was followed by N-butanol extraction yielding 167 mg. Finally, the remaining aqueous layer was collected and dried, giving 198 mg. This stepwise partitioning allowed the crude extract to be separated into fractions based on the polarity, which will be used for further chemical and biological analysis. I performed the thin layer chromatography TLC on the small scale partition sample. Purpose of the TLC was used to compare the chemical profile of small scale partition fractions of EIG and to select suitable solvent systems. As shown in the figure, samples were spotted on the baseline and the plate was developed in the solvent system and capillary reaction until the solvent reached the marked height. The samples are loaded from the left to right as crude extract, hexane, ethyl acetate, and butanol and aqueous fractions, and the separated spots were then observed under the UV light. So this is the comparison of the first two TLC solvent systems. The hexane to EA ethyl acetate ratio of 1:2 shows clear and better separated spot than the 2:1. This means that 1:2 solvent system is more suitable for separating the compounds. The comparison of other two TLC solvent systems using ethyl acetate and methanol. The ethyl acetate to methanol ratio of 1:2 shows better separated spots than the ratio 7:3 solvent system. This indicates that 1:2 solvent system is more suitable for separating these compounds. Next is the comparison of two TLC solvent system using hexane and acetone. The hexane to acetone ratio of 3:2 shows clear spot separation than the 1:4. That means 3:2 solvent system is more suitable for separating the compounds. The conclusion of the TLC analysis of six solvent system shows compound separation depend upon the solvent polarity. Among the six plates, ethyl acetate and methanol 1:2 provide better resolution under UV visualizations and can be used for compound separation and can be applied for a large scale partitioning in the future studies. This is my future work, which includes a large scale partition of the plant sample and then do the thin layer chromatography on the large scale partition sample and then access the cell viability, trap activity, trap staining on the osteoclast cells. Thank you very much for your attention.